This video is about the DPiP lab. DPiP is a man-made chemical that we are going to use in this lab as an indicator to tell us whether or not photosynthesis is happening. The way that we will know that is because the DPiP chemical will change from blue to clear, meaning that if photosynthesis is occurring, our solutions will become more and more clear in color. In order to do this lab, we would typically use a machine called a spectrophotometer, or we call it a SPEC for short, but it's a spectrophotometer. The SPEC holds little square test tubes. We call those cuvettes. The cuvette is about 1.5 inches tall, and it holds liquid just like a test tube. So in each little cuvette, we are going to put some chloroplasts. Our chloroplasts will be coming from spinach, and that spinach was put in the blender just to release the chloroplasts from the cells. And then we are going to put in with the chloroplasts some DPiP solution. That solution in every case will originally be blue in color. For the spinach that we are using 2 sets of unboiled spinach and 1 set of boiled spinach. The hot temperature associated with boiling is going to impact the proteins and enzymes that are in the chloroplasts of the spinach, potentially influencing their ability to do photosynthesis. 2 of the cuvettes will be kept in light, and 1 of them will be kept in the dark. So we will also be studying the impact that darkness has on the ability of a plant to do photosynthesis. So we're looking at light versus dark, boiled versus unboiled. The way that we are going to collect data in this lab is by using absorbance and our machine called the spectrophotometer. The way that the spectrophotometer works is that it shoots a beam of light through the cuvette, and then there is a sensor on the other side that senses how much light was able to pass through that cuvette. Our spec is set to measure absorbance. So if I have a high number on the spec, it indicates to me high absorbance, meaning that there is a dark solution in the cuvette. So that indicates to me that the solution is blue and thus not doing photosynthesis. If I have a low number on the spec, the low number indicates to me that not a lot of light was absorbed, meaning a lot of light passed through my sample, and thus my sample is becoming more clear. So a low number represents low absorbance. In this lab, indicating to me that the solution is becoming more clear, and thus that the solution or the pigments inside there, the chloroplasts, are doing photosynthesis. So what I see in my data chart are some examples of the numbers, so you guys will use those given numbers. You can see that my original absorbance for all 3 cuvettes started pretty much the same. That's only a very slight fluctuation, so they're essentially the same number. And that's because all of my cuvettes started with that blue solution in it. Then, over the course of 15 minutes, I let those cuvettes sit. You can see that those in the dark, the number did not change. So my amount of absorbance, the color of solution in my cuvette, did not change. My boiled on the bottom similarly did not change in number. So my solution is not becoming clear. So only in our middle set of data for unboiled in the light do I see that the number is decreasing. My decreasing number indicates to me that that solution is undergoing photosynthesis. To conclude, the way that this chemical works is that it collects electrons at the end of the electron transport chain. So to remind you, super fast here, in a normal living cell in the chloroplast, we've got photosystem 2, which contains electrons inside of pigments. When those electrons get excited by light, they get grabbed by the primary acceptor and passed down the electron transport chain. There is a second photosystem that also has pigments containing electrons that get excited by light. Those pass down a second electron transport chain. At the end of the electron transport chain, there is a molecule called NADP, and NADP will grab that electron to create a shuttle bus called NADPH. That is going to carry electrons and thus carry energy to the Kelvin cycle to produce sugars. However, in this lab, we are going to use a chemical called DPiP, and DPiP is more strongly attracted to the electrons than NADP. So in the place of NADP, we will have DPiP collecting the electrons. That DPiP will become more clear if it is accepting electrons. So the only way that I will see a clear color is if DPiP collects electrons because these previous steps are occurring.